Coronaviruses are always exploring, and they can adapt and tolerate all kinds of abuse. If we make mutations or deletions in the virus, they still survive. This virus is exploring. We're a very fertile biological playground for this virus right now as a species. There are all kinds of articles written that coronaviruses exceed the theoretical limit of what an RNA molecule can do and survive evolutionarily. They're the biggest, baddest, most flexible RNA viruses out there. Many people thought, well, that's just a problem. These things are impossible to work with. They're so big and complicated. I thought that was fascinating. And I liked them because they broke all the rules. And so no one wanted to work with them because they just kept breaking all the rules. That didn't deter me. Those things enhanced my desire to demonstrate that, in fact, what I'd been thinking about was something that could potentially make a difference in the future. When it gets in a cell, the first thing they do is they rearrange the entirety of the cell. They disassemble their virus and they go in. It's like a truck backing up to a factory with all new equipment, new people going in, rearranging what's in there and starting to make something new. Now, you need someone on the assembly line who's standing and making sure that there's no defects. That's a proofreading enzyme. They look for mistakes so that they don't incorporate problems. RNA viruses don't do that. They're small, they make mistakes and they don't correct them. And in fact, that's probably helped them evolutionarily. So the idea of an RNA virus having a proofreader was not in the textbooks anywhere. It wasn't in anybody's mind. In fact, it wasn't even a concept. So what we did is we developed a system that showed that this function is active in coronaviruses and that they're the first RNA virus family to encode the ability to do this.
Mark is literally one of a handful of people around the world that understand these coronaviruses at that very fundamental, basic level. I'm literally one of a handful of people. And he really understood early on their potential to cause pandemics.
So his discovery in the beginning was people were a little bit skeptical about it. And then quickly people realized it was really important and believed the results. From a theoretical point of view or a virological point of view, it was very important for trying to understand how the virus works, but it also is a target for antiviral therapies.
Mark began not only to study the virus in animals, but also to begin to look at a number of antiviral agents, because certainly with severe infections of coronaviruses in humans, we needed to have better methods to treat them.
I'm pretty sure that there wasn't another lab in the world that was specifically trying to develop antivirals against coronaviruses than mine and my collaborators, of course. They sent us a couple compounds, and they worked dramatically against the wild-type virus, proofreading virus. They really, really knocked it out. We were shocked how well it worked. And I said, I don't know what this is, but can we have some more of this, please? And so they sent us what are called prodrugs, 60 different types, chemical modifications of that drug. And one of those 60 was remdesivir.
You know, early on in this pandemic, you heard stories of a lot of other places that were overridden with patients and, you know, they didn't have enough hospital beds or ICU beds or ventilators. And then when remdesivir data came out and we knew that we kind of had a treatment, I think it instilled hope and it provided hope for many of the providers that, you know, now there's something that we can actively do to care for these patients and try and improve them. So we're seeing from the non-ICU patients, fewer of those are getting sick and coming to us. More of them are stabilizing on the floor and then being able to be discharged from the floor without having to go to the ICU. And in general, the lengths of stay for our intensive care unit patients are also shorter.
There have been places in Florida and New York City where literally we ran out of beds, and there's other patients with other illnesses other than COVID that need to be in a hospital. And if a hospital has no beds, the ability to get patients out of hospital quickly is also important because that creates beds for new patients. And so remdesivir clearly decreased length of stay. Once patients end up on a ventilator, the mortality can be as high as 50 or 60%. So we absolutely have to stop patients getting to that stage.
What seemed like sort of a miracle drug that was sitting on the shelf ready to go in 2020 when COVID emerged was really the basis of work that probably started back in 2007 but really took off in 2014. So it takes time, and it takes commitment to the process. You can't just do it overnight. When we have our lab meetings and everybody is feeling anxious, I say, this is way too urgent for us to hurry. On the flip side of it is, this is the most rapid progress ever in history toward vaccines and multiple compounds.
Mark's lab is also working with the vaccine companies to actually show that their products are effective in producing antibodies, which are actually active at killing and preventing replication of the virus.
I was vaccine volunteer number 1 for the Moderna trial here at Vanderbilt. That was very exciting.
He shows great humility, and I think that's a great trait for building teamwork because Mark sees himself as a team member. He doesn't see himself as some boss at the top of a team.
The first thing when I think about Mark is he's a Renaissance man. Like, he can do pretty much anything.
I will spend as much time doing photography and working on Photoshop and playing penny whistle and doing woodworking as I will doing science. So I think there's that kind of creativity. There's also parts of science that are just pure beauty. If you're sitting at the microscope or you're watching this in time lapse, it's just beautiful. It's like watching a supernova or a cosmos explode, and it's nothing except for sort of an existential ecstasy. It's fantastic. We feel like frontiers are all gone, so I guess I've chosen to dive into the nanocosmos. Right? I've chosen to dive into an area where there's an infinite frontier that gets smaller and smaller and seems more detailed. I like new ideas. I like new things. I like to test beyond what we're thinking about and say, why are we thinking of it this way?
He has a good moral core. He has a basis that he believes that everyone has worth, that everyone should be listened to, that everyone has something special to give. And I think that is kind of the basis upon which he operates. And obviously, I find that really refreshing and helpful.
No matter what he says, he always does what's right, and he shows up when you really need him to show up.
I hope that it leads to an opportunity to have a voice, I call it, I guess, a voice of authority that says, listen, this is real, that it's not supernatural, that it's not an opinion, that there's really data and facts and there's science behind it that we can understand. There's something we can do to intervene.
So I think there's a paradox. Nobody wants there to be a pandemic to prove the validity of their science or their work. But having said that, if you're faced with a pandemic, you absolutely need the Mark Denisons of the world to be there and ready to step up.